Vaccines can be defined as inactivated or weakened pathogens. Vaccines are broadly classified into 6 different types. First one is inactivated. Second, it is attenuated. Third, it is toxoid. Fourth, subunit type. Fifth, conjugate. And finally, we have the heterotypic type. So we learn one by one each of them. The first one we learn is inactivated one. Some of the examples of inactivated one is polio, influenza vaccines. In this case, a previously active pathogen is being made inactive with certain agents such as chemicals, heat, and radiation. So they make it inactive. The next type is the attenuated vaccine. In attenuated vaccine, we mostly have viral vaccines. For example, we have MMR vaccine, which is basically a combination of measles, mumps, and rubella vaccines. Here again, what we are doing is we inactivate the viruses. The way they are being cultured itself is being made them inactive. But in certain cases, we also have bacterial vaccines in here, such as tuberculosis vaccine. These are certain examples under attenuated vaccines. In toxoid form of vaccine, we are only taking the illness causing substance. So, we are taking an inactivated toxic compound from the microorganism. So, from microorganism we are taking the toxic compounds. So, example under this is tetanus and diphtheria vaccines. Under subunit vaccine, we have two examples. One is human papilloma virus vaccine and influenza vaccine. Now, in this case, we are going to use the technique of recombination. We are not going to take a completely inactivated microorganism or attenuated microorganism. Rather, we are just taking a part of it, a subunit of it. This is why we call it a subunit type of vaccine. We are just taking a small part of it and say combine it with, conjugate it with say yeast or algae or something of these sort, and then they are being introduced. In the similar manner, even plague vaccines are also being created. Next vaccine is conjugate vaccine. In here, we use the polysaccharide which is present around the coating of the bacteria in the form of form which generally is a immunogenic substance. These substances are the one which is being extracted and then they are being used as the vaccine. Again, the example here is influenza type B type of vaccine. With that, we are left with the last one, that is heterotypic vaccine. Heterotypic vaccine is also called as Jennerian vaccine. The reason we call it this way, because for the first time Jenner had used this. So Jenner, what he did was he used cowpox extract to cure smallpox. So what we are basically doing in this heterotypic vaccine is a different animals similar type of disease extract is taken, which either does not affect at all or does not cause any kind of disease in human, or even if it cause, it will be very very mild. So that's something which has been introduced into the body, and once it has been introduced, the person is kind of immune to the bigger disease.